All right, future officers, listen up. If you're prepping for that NDA exam, you know general science is a make or break section. Today, we're zeroing in on one of the most fundamental topics, the cell and cell division. Let's get your mission ready.
So let's kick things off with a big picture question. What is that one single idea, that fundamental building block that connects everything from a microscopic bacterium to a person?
You got it. It's the cell. Every single living thing is made of them. I want you to think of it as the smallest possible packet of life. It's got all the machinery it needs to function, all wrapped up in a nice little package. For your exam, this is ground zero.
Okay, here's our plan of attack. We're going to start with the blueprint, cell theory itself. Then, we'll cover the two main types of cells, take a tour of the cell's internal machinery, zoom in on the command center, the nucleus, and finally, tackle how cells actually divide. Let's move out.
First up, the blueprint of life. Now, understanding cell theory isn't just about memorizing a few facts. It's about really getting the entire framework of biology. You know, our understanding of this was built up over centuries. It all started way back in 1665 with Robert Hooke. He looked at a piece of cork under a microscope and saw these little box-like things he called cells. But it took nearly 200 more years for Schleiden and Schwann to figure out that, hey, all plants and animals are made of these things. And then Rudolf Virchow came along and delivered the final, crucial piece of the puzzle.
And that final piece, it came in this powerful little Latin phrase from Virchow in 1855. Omnis cellula ad cellula. It just means all cells arise from pre-existing cells. This one idea completely destroyed the old theory that life could just appear out of nowhere. It proved that life only comes from life. That was a game changer.
So, these 3 points are the absolute core of cell theory. You need to burn these into your memory. 1. All life is made of cells. 2. The cell is the most basic unit of that life. And 3. Cells only come from other cells. It's simple, it's powerful, and I can almost guarantee you'll see it on your exam.
All right, let's move on. Now we're going to look at the two basic designs for life on Earth, prokaryotic and eukaryotic cells. Knowing the difference between these two is a classic exam question, so pay attention.
First, you've got the prokaryotes. These are the originals, the veterans of life on Earth. They're super simple, really small, and here's the most important part. They do not have a true nucleus. Their genetic material just kind of floats around inside the cell in a region we call the nucleoid.
And then you've got the eukaryotes. That's us, and every plant, every animal, every fungus you've ever seen. These cells are way bigger, much more complex, and their defining feature is that well-organized nucleus that keeps all the DNA safe and sound. Plus, they're packed with all these little specialized compartments called organelles.
Okay, this slide right here, this table is high yield for your exam. It lays out the key differences perfectly. Just look at the size difference. Look at how the nucleus and DNA are handled, and see how eukaryotes have all those complex organelles. Even the ribosomes are different, 70S versus 80S. These are exactly the kinds of details that get you those extra points.
So now that we've got the eukaryotic model down, let's take a tour inside. The best way to think about a cell is like a busy factory. Every little organelle is like a specialized department, making sure the whole operation runs without a hitch. Here's a quick-fire round of the major players. You've got the mitochondria, the powerhouse, generating all the energy. The endoplasmic reticulum, that's the main factory floor, making proteins and lipids. Ribosomes are the actual machines doing the assembly. Lysosomes are the cleanup crew. And the Golgi apparatus, that's the shipping department. They all work together, right? The ER makes the stuff, the Golgi packages it, and the mitochondria powers the whole show.
Now, even within eukaryotes, there's another key distinction you gotta know. Animal cells versus plant cells. This table, another exam favorite. The big 3 differences you absolutely have to remember are that plant cells have a rigid cell wall, they have plastids like chloroplasts to do photosynthesis, and they have a huge central vacuole. Animal cells don't have those, but they do have centrioles, which are important for cell division.
All right, let's zoom in on the most important department in that whole factory, command central, the nucleus. This thing doesn't just sit there. It is actively controlling everything the cell does because it holds the master blueprint.
So inside that nucleus, the genetic blueprint isn't just a tangled mess of spaghetti.
No, it's neatly packaged into these structures called chromosomes. And each chromosome is basically one incredibly long molecule of DNA that's been coiled up super tightly around proteins. This organization is absolutely key to making sure that information gets copied and passed on perfectly.
Now, the blueprint itself is written in two related languages, DNA and RNA. Think of DNA as the master copy. It's a stable double helix that stores all the genetic info for the long term. RNA is more like a temporary message, a single-stranded copy used to actually go out and build proteins. And notice that key difference in their chemical bases. RNA uses something called uracil, or U, where DNA has thymine, or T. That's a classic exam question right there.
So, we've got the blueprint, we've got the machinery, but how does one cell become two? That brings us to our final and maybe most important section, cell division. This is the process that drives everything from growth and healing wounds to reproduction itself.
Before we get into the nitty-gritty, let's just lock in one key number, 46. That is the diploid number, the full set of chromosomes in a typical human body cell. You get 23 from your mother and 23 from your father, 46 total. Got it? Good.
Okay, the first type of division is mitosis. This is what your body uses for growth and repair. It's a really elegant four-step dance. In prophase, the DNA packs up tight. In metaphase, the chromosomes meet in the middle. In anaphase, they get pulled apart. And in telophase, you get two new nuclei. The end result? Two new daughter cells that are perfect clones of the original.
But for reproduction, you can't just make clones, right? You need to combine the genetic info from two parents. And to do that, you first have to cut the chromosome number in half. That is the very specific job of meiosis. Its whole purpose is to create sex cells, or gametes, like sperm and eggs.
And here it is. This is the ultimate comparison slide. This is pure gold for your prep. Mitosis, one division, two identical diploid cells. Meiosis, two divisions, four genetically different haploid cells. And crucially, see that crossing over point? That's where chromosomes literally swap pieces of genetic material. This is the engine of genetic diversity, and it's why we're not all identical.
So, why does all of this matter? What's the bottom line? Well, without cell division, nothing could grow, nothing could heal, and nothing could reproduce. Mitosis builds and maintains our bodies day in and day out. Meiosis makes sexual reproduction possible and makes sure the chromosome count stays stable from one generation to the next. It is literally fundamental to the continuation of all life.
So let's end with this thought. This whole incredibly complicated process is governed by a set of perfect, precise controls. But what happens when that control system breaks down? What happens when cells just start dividing uncontrollably? Well, that question leads us directly to diseases like cancer, and it highlights just how critical this biological machinery really is. Keep studying smart. You've got this.